Hello, dear friends. Welcome back to my YouTube channel. I'm Nitin Naik. Today, going to represent a lecture on approaches to isolation and purification of microorganisms. So let's start. So in this lecture, we are going is what are the methods for isolation of pure culture, what are the methods for maintenance of stock culture, and what are the methods for systematic study of pure cultures. So there are main three methods being used for isolation: strict plate, pour plate, and spread plate. Then this culture, once it is purified, that is maintained by either agar slant method or agar step method. And the purified culture is studied by using morphological, cultural, biochemical, serological, and genetic characteristics. So these are the three methods, that is strict plate method, pour plate method, and spread plate method are used for the isolation of pure culture from a mixed culture. Okay. So in strict plate method, the sample is strict on a nutrient agar or a basal medium, and as the stricking is carried out, the load of microorganism is minimized to get a single colony, which is called as well isolated colony, and that well isolated colony which is further purified, again stricking, and then that purified organism is grown on a specialized media for its identification. Or second method is a pour plate method. In this, the culture or mixed culture in the form of broth is spreaded or poured over the pour in a plate. Above that, the medium is poured and incubation is carried out, and after incubation the colonies are observed. There are some subsurface colonies are observed, there are sub colonies which are present on the surface, because the firstly the suspension is taken up, and after over that the media is poured. That's why the both type of colonies is studied. This is the advantage of pour plate method. And in spread plate method, the media is uh, solidified on a plate. After on that media, the culture is okay, 1 ml of culture is taken up, and that culture is spreaded by using glass spreader. And after that the incubation is carried out, and as after incubation the well isolated colonies are selected, and these colonies are further taken up and they are purified. So this is the method for called as according to spreading. This method is called as spread plate technique. Here, according to pouring, this method is called as pour plate technique, and according to streaking, here only streaking is carried out. That's why this method is called as streak plate method. So these are the methods for isolating pure culture. Another method is the serial dilution method, where the sample is taken into first or there is a series of 9 ml distilled water is carried out, and serially transfer of subsequent transfer of the culture from one tube to another tube is carried out, and to minimize the load of microorganism. Hence this method is called as serial dilution method. And the serial dilution sample, serially diluted sample, is used for pour plate or split plate technique. So if you see this sample, whatever it is taken up, it is serially diluted. This is also serially diluted sample. Next, what is a pure culture? A culture in which only one strain or clone is present is called as a pure culture. And what is a mixed culture? A culture which contain a lot of organisms. So this is a mixed culture. If you look out, there are different types of colonies you are observing, and this is the pure culture where only single colonies there. Once one can get the pure culture, it has to maintain, because the maintain a live strain, to maintain the strain uncontaminated during study, to maintain the strain stable for longer period, and stock. So these are the main objectives for which the strain is stored or maintained, and this maintained by either agar slant, means on agar slant, or in agar step. After the strain is purified or isolated, its morphological characteristics are studied, its cultural characteristics and its biochemical characteristics are studied. So what are the morphological characteristics? The first is the gram nature. It includes its size, shape and arrangement of the cell. And the motility includes the study of presence of special structures like flagella. So based on gram nature, there are two groups of microorganism, as you know. One is a gram positive and one is the gram negative. The method discovered by Christian Gram. The motilities depends upon the flagella. There are two groups of the microorganism. One is the motile, one is the non-motile microorganisms. The cultural characteristics include the colonies on solid media. If the solid media is used, then the colony is there. There is group of cells, its size, shape, color, margin, elevation, opacity and consistency. Also, if the colony is grown on liquid media, they have different types of characteristics. If grown on agar slant, there is also different type of characteristics, cultural characteristics. So let's talk the cultural characteristics of bacterial colony on agar surface. The shape from circular, rhizoid, irregular, filamentous to spindle. So these are different shapes one can observe on a solid plate. Margin of the colony entire, undulate, lobate, curled, rhizoid or filamentous. So these are the different type of margins may the colony will have. Then elevation ranging from fulminate to umbonate. So these are the different type of level is small and large. So one of the size your microorganism smooth or rough, dry, mucoid or rugoid. So these are the different types of textures are also one of the texture. Then appearance, that is glistening, shining or dull, and pigmentation, that is non-pigmented, that is cream color, tan color or white color colony, or if pigment is there then purple color, red, yellow color like colony. An optical property, it is either opaque, translucent or transparent. So your colony may have one of the shape, may have one of the margin, may have one of the elevation, one of the size, one of the texture, one of the appearance, pigmentation from this and optical property on all of above. So based on your this all characteristics, the identification, partial identification of microorganism is carried out. These characteristics or these results are matched with the Buggy's manual of systemic pathology, and then the identification of microorganism is carried out. If the colony is grown on a observation slant, it forms a filamentous or thread-like growth, otherwise a arbuscent growth, or that is tree-like growth, or braided growth, effusing or spreading growth, or rhizoid growth, rhizome-like, and ethionolate or spiny growth. So these are different types of cultural characteristics which are observed on a agar slant tube if you are working the organism on a slant. Thank you. So these are the, and after that the biochemical characterization like catalyst test, H2S test, and so many tests are there that are carried out for the ability of microorganisms to utilize different types of sugar, ferment sugars, or production of some gases like amino acid decarboxylation, amino acid deamination, urea test, and so many tests are there. So this is all about isolation and purification and observation of the microorganisms. Thank you. If you like my video, click on like button. Don't forget to subscribe my YouTube channel by searching Nitin Naik, and please click on bell icon for more upcoming such videos. Thank you.